Okay, so we are back. It's episode number 6 of Coronavirus Conversations. I'm Adam Hertzberger, and with me is my colleague, Dr. Josh Williamson of the Gable Health Center at Albright College. So for those of you who have been regular viewers, thank you for viewing another episode, and hopefully we'll have some new people that will sort of tune in and get some awareness and information about the pandemic, and we'd love to hear additional questions that you might have. We'd be happy to take any questions and use it as a point of discussion. Okay, so Josh, so why don't we start? Are there any sort of clinical updates or anything that you wanted to bring up since the last time that we spoke?
Sure, Adam. Yeah, it's interesting, Adam and I get to have a lot of conversation before we start the episodes, and it's always great. There's so much stuff out there, and I'm sure everybody is doing the same thing, kind of being surprised each day by additional information that's out there. Some of it's really good, some of it maybe not so good. We definitely all have tons of questions that we hope to get answered. The World Health Organization set up what they're calling a mega trial. It's called Solidarity, and basically they're looking to enroll patients with confirmed COVID, and then they're going to randomize patients to either standard of care, which does vary from place to place. So it'll be interesting how that plays out once they get patients enrolled. And then one group will be randomized to remdesivir. One group will be randomized to chloroquine, hydroxychloroquine. And then the two other groups will be either given lopinavir, ritonavir combination, or lopinavir, ritonavir, and interferon beta 1A.
It's a lot of moving parts there.
Yes. And I hope that I didn't mispronounce those medications too badly. They're not ones that I'm prescribing.
No, I think you did a good job.
Do you want to talk about those? We talked a little bit about remdesivir before and chloroquine and hydroxychloroquine, but those other two I don't think we touched on too much. Adam, do you want to?
So it's interesting. What's interesting about this trial, Josh, is I'm trying to keep all the drugs straight in my head that you just mentioned. All of the drugs that you mentioned, except for the hydroxychloroquine, actually are thought to operate on a viral enzyme. They're basically a viral, inhibitor, whereas the hydroxychloroquine is thought to modulate, I guess, more the immune response. I mean, there is some in vitro evidence, and this is a bit of a review from some previous episodes. We did talk, as you mentioned, about the chloroquine a little bit. I mean, if you take SARS-1, and I'm sure people are doing this for SARS-CoV-2, but I haven't seen it actually published yet. But I know for SARS-1, if you treat cells in vitro, meaning in a plate of cells growing in a lab, if you treat cells with chloroquine and then infect with SARS-1, it will inhibit the infection. And it's thought that it actually raises the pH. So the virus needs, when it's going into the cell, the pH of the compartment that is in drops. So it becomes more acidic. And it needs that acidic environment to basically, in this, I'm trying to put this in sort of, non-scientific jargon, but it basically needs that drop in pH to sort of actually enter into the sort of interior of the cell and start to replicate. It doesn't sort of fully mature, if you will. So hydroxychloroquine is thought to keep the pH closer to neutral, say closer to 7, which is what is sort of like water is neutral or just slightly basic, or even sometimes acidic for that matter. But anyway, so it's thought to inhibit that entry step. And that's honestly unclear to me if that's what's happening in the body, because as we discussed, hydroxychloroquine is also given to patients with inflammatory disorders and autoimmune disorders like lupus or arthritis, I guess, in some situations. And so is it modulating the immune response, or is it modulating the virus? Is it both? That's unclear at this stage of our understanding. But like I said, if you go back to this idea of this overactive immune response causing the disease, then perhaps if the hydroxychloroquine, if given a little bit later in infection, maybe that might help modulate that immune response. But that's kind of unclear. So, Josh, did you want to?
And that's a great review there, Adam. Thank you, because that is very interesting to me. Basically, in medicine over decades and centuries, people try certain things and they seem to work. And then they run through their massive studies to show that. And sometimes it works, but it's not always the theory is incorrect. The medication still works, but then it's kind of rebounded later, and they say, oh well, no, this works because of this reason. So it's amazing that we know all these little details, but it's also amazing to me how much more we're going to find out, in 10, 20, 50 years from now.
Exactly, because this will be studied for, as you said, years.
Adam, I think you may have grown a tail there for a second. I saw.
Yep, this is we have our little visitor. This is Stella. So she was sleeping on the bed, but now she wants to make an appearance, I guess. So the other drugs you mentioned, the remdesivir, we did discuss that already. So that was made by Gilead, and that was tried for Ebola virus. The results were kind of mixed. It really didn't show much of a benefit, but they already kind of had it on the shelf, and it did show some in vitro activity. Once again, in the lab, it showed some in vitro activity against the seasonal coronaviruses and even SARS-1, I believe. So they thought, okay, well, let's try this for SARS-CoV-2 in people. And then the other drugs you mentioned, those.
And Adam, I just want to say about that, we were really timely on that. I think if we go back on the dates of our recording, we were that was leaked. that study information was leaked early, and in fact, I think in the initial pieces I read on that, I think the death reports were actually flip-flopped. I'd like to go back to try to see that again, but
yeah, can you just remind our listeners about that a little bit? Like, what do you, just to kind of explain?
Yeah, so basically, they said it was leaked because the results of the study were announced to the press before they were announced in a scientific forum. So a lot of experts were upset by that. The rationale for the leak, from what I'm told, was that because there was benefit, they wanted to get the word out immediately as fast as they could to everyone, so that so patients that were on the placebo arm of the trial could be switched over to the medication. Whenever you see a benefit in a trial early on, it's unethical to withhold the treatment from the placebo arm of the study. So you basically end the study and say, hey, this works, let's use it. So that's what happened with that. The medication was shown to shorten hospital time very significantly. And I think I said that there was no change in death rate because I think those numbers were actually flip-flopped.
Okay.
I think it did. I think it still was reported that it wasn't statistically significant. Sorry, but I think they were reporting it as trending towards reducing death rate as well.
Gotcha.
It's thought that if you give the medication earlier, it's better.
Right, and that's a theme that I just want to bring up again. That just from a purely scientific viewpoint, if a drug is going to target the virus itself, then probably the earlier the better, to try to dampen or reduce the levels of virus replication. Based on what we're seeing, anything that targets the immune system probably, and once again, this is just, a sort of educated viewpoint without really any medical, sort of expertise to back it up. But it would make sense that you don't really want to dampen that immune response too much in the beginning, because you're using that to fight the infection, but you want to try to reduce the cytokine storm, if you will, that overactive immune response that some people get during that like second into the third week. So to target the virus early, but probably the immune system a little bit later. And that's just kind of like my gut feeling at this point. And it's still unclear. People who do mount this overactive immune response, it's still unclear why. So people are going to be starting to study, are there genetic factors, for example, are there what are the factors? Can we almost predict, right, based on X, Y, Z? Can we predict who's going to decompensate, who's going to have that severe immune reaction that becomes uncontrolled? Is there some kind of like I'm trying to think of the right word to use. Is there some sort of indicator, like a warning sign that, oh, this person. There's one thing I just want to interject as well. So I've been following some stuff, like I said, by this. His name is Dr. Daniel Griffith. He's an infectious disease doc in New York City, and I've been following some of his updates. He's like literally in the trenches with these patients, and they're working on publishing a study of roughly, I think a few hundred patients, and he seems to think that what's called the neutrophil to monocyte ratio as being predictive of disease severity. And so basically a neutrophil, for those of you out there, it's the most abundant white blood cell that is floating around in our bloodstream at all times. It's really important for fighting off bacteria. There's potentially some antiviral capacities, but this is one of these cells that it's called a phagocyte, that they literally go around and engulf or ingest microbes, and they're thought to be really, really important for fighting off bacteria. But the problem with neutrophils is once they get activated, they are very dangerous cells. They can kill both healthy tissue and the microbes. So these are cells that are not, they're very short-lived, and they're kept in the bloodstream in an immature form until they're kind of called into action, because you really have to be careful with these activated neutrophils. And Dr. Griffith noticed that in patients that tended to do worse and have more severe immune responses, they tend to have a much greater abundance of neutrophils compared to patients that recovered faster. So it's one indicator potentially. It needs to be borne out in a larger study, but that's the first kind of indicator that I've seen out there, this overabundance of neutrophils. And these neutrophils are going to be releasing a bunch of these inflammatory molecules and potentially making things kind of worse. Basically almost like too much of a good thing, right? Immune response is a good thing to fight an infection, but within reason. It has to be regulated. So,
and that's that's done in other scenarios also, Adam. For example, babies, the immature to mature white blood cell ratio is an indicator of severe infection in newborns. So that's something that has been looked at in other conditions as well. So kind of.
Maybe that's where they got the idea to look for it. Maybe, yeah.
Yeah, I'm sure they've been looking at all those different inflammatory markers. I know there was I've read some stuff about troponin as far as indicator whether the heart's affected, which is a big concern and a big topic in medicine. How badly this affects heart muscle.
Sure.
Are people that recovered going to have problems down the line because of those effects? It's interesting, but yeah, anytime you can find a good indicator that these are the patients that are more likely to get sicker, then we're much better off.
Now, I just want to mention the one issue with remdesivir is that needs to be injected. It's an IV drug, so that could only be administered in a clinical setting. It could theoretically be given at even like a patient first. They had the expertise to put something into a vein, but just so people are aware that that's not a pill. Now the other drugs that Josh mentioned,
you weren't plugging that company.
No, I'm not paid by patient first, no. But I'm just saying, like, you don't have to go to the hospital, luckily, for something like that. But once again, these are all not FDA approved as of yet. These are all still clinical trials. So the other two drugs are already FDA approved. Those are the, try to say this again, that's the
Lopinavir and the
lopinavir and
ritonavir.
ritonavir. Yeah. All right, so just so people are aware, a little bit of virology background for this particular part. So those are licensed HIV drugs, and HIV, when it replicates, it during its life cycle, it makes a long, what's called a polyprotein, and it's basically a long protein, but it needs to be chopped up into individual segments, and each segment then becomes an active protein in its own right to, become parts of the virus and to become a fully mature virion. And so with that, if you inhibit that enzyme, it turns out it's a viral enzyme it's just called protease. That cleaves the protein. So protease just means that it clips up a protein. And so if you inhibit that viral protein, you inhibit the virus. Well, it turns out that coronaviruses also make something called a polyprotein. It makes a long protein that it has to clip up, and it has a viral protease just like HIV. Now, it's a different protease. It's not exactly, it's not highly related. But I think the idea behind that, Josh, is that they're kind of almost hoping in a sense that it might cross-react with the coronavirus protease. And even if it cross-reacted a little bit or had a little bit of inhibitory activity, that perhaps that could be the difference between life or death, even if it worked a little bit. But just so people are aware, those two things are HIV drugs that they're trying to kind of repurpose as potentially coronavirus drugs. Once again, probably if it works, given earlier the better would be my guess. Okay, so yeah, so we'll wait the results of those trials. I'm also, by the way, Josh, I have been looking for results from some of these early vaccine trials. I have not seen anything come out as of yet. There's actually also a growing rumbling from some vaccinology people that perhaps younger people would actually volunteer. I mean, they would be compensated, and it would certainly be a liability issue, but actually sign up for a challenge study to try to further the vaccine process along faster. It would definitely go along faster if you could vaccinate, say, wait, I don't know, 15, 20 days, whatever, then actually challenge with SARS-CoV-2 and see what happens in those patients. The only other way to do a vaccine trial, and honestly, these drug trials as well, is you have to wait for people to get sick, and then you give the drugs, right? Or give the vaccine, and then you just track people, do they get infected less than the group that didn't get the vaccine? And of course, that process takes longer than just vaccinating and then just challenging people with the virus. So
and a lot more patients, right, Adam?
And a lot more patients, right? So you would need less patients for a challenge study because you knew exactly how much virus they got. You could control when they get infected. Whereas like you said, Josh, right? If it's sort of vaccinate and then wait to see, you theoretically would need more patients to potentially look for a positive effect. So yeah. Okay, so that's the recent WHO trial. All right, so we'll have to wait on that. So, okay. Were there any other clinical things that you've heard from the last week that you wanted to bring up?
No, I think that was the most interesting one. There's I think I counted up to about 30 different treatments that, that's obviously probably the biggest trial that's out there, but then there's literally dozens of smaller trials that are happening, looking at a host of different medications. We've kind of touched on some of those along the way, but again, like you said, basically repurposing medicines that are out there to try to see if they're effective for this virus, as opposed to what they're typically used for.
Right.
And then the vaccine trials, I think there's about 20 of those going on.
There's about 20 to 25 at this point, yeah. I mean, the advantage of sort of repurposing is at least those trials that are already licensed, or those drugs that are already licensed, at least they've gone through a lot of the safety testing already. So that is one nice thing about doing something sort of quote unquote off label, is it's been licensed for this other thing, but it's already been tested for safety. So at least that's one less thing to have to kind of worry about in like the pipeline, because usually these trials, they look for safety first in a smaller group of people. Of course, it's been in animals, right? It's been in mice and, larger animals past the safety testing there. Now we're going to try it in a small group of people, because you mentioned about like sort of ethically, you can't enroll, say, a 500,000 people trial without doing a smaller scale safety trial first. Because what if there's all these complications or side effects and you're giving it to all these people?
And basically, that's all these medication commercials where they list the side effects. Those are side effects that they knew about early on, and then they're also post marketed. So once the medication is released and you do have millions of doses going out, then they add on those extra ones that they see afterwards.
That reminds me of what, like, Vioxx is probably a famous example of that, right? It was licensed, it went through all that testing, and then there were some complications that arose well after that when it got out to the general population, right?
Yep.
Yeah. Okay, so yeah, I guess so. The only other thing I wanted to bring up in this particular episode is it's sort of an interesting scientific question, and one of the things that is sort of, as a scientist, you always want to have like all your ducks in a row, and it's hard during this pandemic because people want answers and potential life-saving treatments as fast as possible. But sometimes I think the scientific process has been a little bit compromised, either willingly or unwillingly, in this rush to get information out there. And also the media needs to do a better job of kind of parsing out some of the details and worrying less about clickbait than like a thorough analysis of a study. And so using that as a preface, there's been some recent reports, and I wanted to bring this up, of a potential mutation, and the one that's getting the most press it's called D614G, and I believe D is isoleucine. I'm trying to think back to my biochemistry class. D is just the one letter abbreviation for an amino acid, and I think, it might be aspartic acid. And anyway, D is an amino acid, and it's being changed 614G. G is a glycine, so it's changing the amino acid in the spike protein, so the protein on the outside of the virus. And there's been some reports, and this all came from one of these preprints. And like I said, preprints can be really good because it gets information out there, but it's also not peer reviewed, so you have to take all of that with a grain of salt. So, I mean, it's literally like almost like writing an editorial. I mean, anybody can write a preprint and post it on the preprint server. So it hasn't been reviewed by experts in the field and been published yet. So anyway, so they and the media has said, well, this might increase transmissibility. And I just want to say from a purely scientific perspective, that is not substantiated at all. I read that particular preprint. I've read some of the media about it, and I think part of it is, and Josh and I were talking about this earlier, is some of it could be sampling bias. So if you're only sequencing virus from the sickest patients, or from even like a hospital setting, right? I mean, let's say, or like a nursing home, you're in close confines, people, in close contact, even if you're wearing masks and gloves, there's still a higher risk of transmission because of the nature of all these sick people, close confines, all this stuff, right? So if you see this mutation in that setting, it could have nothing to do with the ability of the virus to spread. It could solely be from sampling bias, that you're sampling in this particular setting. The other thing I just wanted to mention as a virologist is that there's something called a founder effect. And there could be just a random mutation happen, and that virus, where that mutation occurs, spreads to a new population. And now that virus starts to spread in that population. Well, that's called the founder effect, because that mutation was there from the beginning, and then of course every virus after that is going to have that mutation. So once again, it's almost like a passenger, right? It's sort of this bystander, this passenger. It's there, it's spreading, but it's only there because it was there to begin with, and everything else came from it. So by definition, it's going to come from that initial virus isolate. So it's way too early to say that mutation, or really any mutation you read about, is having any kind of effect on the disease. So I just wanted to make sure that people understand that, that are listening.
And I think the other point to that, Adam, like we talked about before we started recording today, was that that mutation was identified a while ago.
Correct. Yeah.
And so there is an interesting website where they kind of show where, the thought that, obviously we still think this came from Wuhan, China, spread to Europe. Well, that mutation spread back into China.
Okay.
And so they've been following that along with, other small mutations, because isn't that right, Adam? As if, like, say I get infected with the virus and I pass it on to someone else, there's those little pieces that basically came from me that are now part of what's going on to other people, and they can somehow, you guys are able to tease that out and follow that. Is that right?
Yeah, pretty
Am I
much.
understanding that correct?
Yep. Right, because the virus has to start from from what it had at the start, right? I mean, so it's going to have those that genetic signature of what it started with. Now it could change from there, but at the end of the day, it's going to start with that initial founder population, and it could either stay the same or it could change further. But like I said, that mutation could be there just as sort of a passenger, just as a bystander. It has nothing to do with anything other than the fact that it's just another part of the sequence of the virus. So anyway, this is also a good point to mention. This hasn't been brought up yet because I'm trying to like sort of pepper in some of the virology as we go. This particular virus is very interesting just from a purely biological standpoint. It's actually the largest of all the known RNA viruses. So remember, this has an RNA genome, and other viruses that have RNA genomes, such as flu, HIV, Ebola, those genomes tend to be about 10,000 base pairs. This one's about 40, like roughly. I'm kind of rounding. It's roughly 40,000. So it's about 4 times larger, which is quite a bit. And the reason it's able to get bigger is this virus was discovered about a decade ago. When it's copying its genome and making more genomes to make new virus, it actually has what's called a proofreading function. So if it makes a mistake, if it makes a mutation, often it will actually correct it to put it back to what it should be. So this virus, compared to HIV, compared to flu, compared to pretty much all other known RNA viruses, it actually has a lower mutation rate than these other RNA viruses. Influenza, if you think about it, we need a new seasonal flu cocktail every year because of how easily it can mutate. Whereas all signs so far for coronaviruses, it is mutating far less frequently. There's been some estimates that so 40,000 bases, right? So the genome's roughly 40,000 what are called nucleotides or base pairs. If it's been speculated that because it mutates less frequently, because it has this proofreading, that during the course of an entire year, it's been speculated it might make about 20 nucleotide changes, which are called mutations, right? So if you think about it, 40,000, and if in a course of a year, it's been estimated that it can mutate 20 times, that's actually pretty low mutation rate. That's actually very low, for a virus at least, or especially an RNA virus. So this is not mutating rapidly at all, just because of its biology. And so that gives me hope as a scientist that if we can find a vaccine that, say, makes antibodies that then target, say, like one of the proteins, like the spike protein, it's not going to mutate as rapidly as other viruses will, such as HIV. And so it gives me hope that if we can design a drug or design a vaccine, it's going to be not quite as easy for the virus to just simply mutate away and be like, okay, I'm done with you, immune system, or I'm, whatever, like, that's the best you got kind of thing, it's not going to mutate as rapidly or as efficiently because it has a proofreading function. So
We're almost out of time, Adam, but
yeah,
are RNA vaccines more difficult to make, or do they
against
work
RNA
as
viruses,
well?
you mean?
Yeah, yeah. Vaccines against RNA viruses, are they more difficult to make? Are they less effective in general? Do you want to comment on
It
that, or
just
should we save
depends.
that for another time?
It just depends. I can give a brief comment. We can start up in the next episode on this point. Um, it really depends. So some RNA viruses mutate less frequently for reasons that have nothing to do with, with proofreading. It gets a little bit complicated, but so for example, measles, right, is relative, and so the term is antigenically stable. So the antigen is what our immune system targets, and if that antigen doesn't change a lot, it tends to be easier to control that pathogen because you make an antibody, boom, it's inhibited, and it's not going to change it to something else. So like something like measles, we've had pretty much the same measles vaccine for decades, and it's a very good vaccine. But it turns out measles is relatively stable with respect to its antigens. Same with like polio, relatively antigenically stable. There's only 3 strains of polio, and 2 have been declared eradicated. Whereas you take something like HIV or flu, they just, as part of their natural biology, they mutate very efficiently, and they mutate a lot. So I don't think there's a coincidence that it's hard to have a good vaccine against flu. Um, and it's actually, at this point, we have no vaccine against HIV because HIV mutates even more efficiently and rapidly than flu does. So, the more you change that thing the immune system recognizes, the harder it is to control. So like I said, luckily for coronaviruses, all evidence points to it's going to be more like a measles, where if you have a vaccine, it's going to be stable, easier to target, luckily, because there hasn't been, honestly, a lot of good news for coming out of this pandemic so far in terms of how to treat the most severe people, right? But that is some good news, I think, that's going to come out of this, that it is antigenically stable. It doesn't mutate rapidly. And like I said, HIV is probably the hardest thing we as a scientific community and a medical community, the hardest thing we have ever had to try to deal with. We don't have a vaccine. There's not even one close, as far as I know, because it can mutate so rapidly and so much. So yeah, does that kind of
Thanks
adjust
for
what
that.
you're thinking, or?
Yeah, yep.
Yeah. So there is kind of a silver lining with the fact that it does have this proofreading. It doesn't mutate as much as other RNA viruses do. So there's some hope there that a therapy will work. Great. Well, did you, like, we have I have like a minute or two. Do you have any kind of closing thoughts, Josh? Anything you wanted to convey while we're here?
Um, well, I guess just a quick, I don't know, I've been in a lot of meetings that are talking about how we're going to manage kind of as we start to reopen in Pennsylvania and how things change. And there is a lot of things that we need to deal with. But on the flip side, the good news is that in the United States, in Pennsylvania, in the Northeast, we're trending down in this pandemic. And I think there's some hopeful news out of the Southern Hemisphere. People are talking about how Australia and New Zealand are doing well as they're heading into their winter season. So that hopefully will give us some glimpse into how much our fall and winter season, once we hit flu season again, where we'll be as a nation. Um, but I personally think that hopefully we're past the worst of this, and hopefully we don't see a big resurgence. But only time will tell.
Yep. Hopefully we don't see it. Hopefully we have at least a vaccine. So yeah, So let's start episode number 7. We record next week. Let's start with another recap of this, and I think we'll go into some. You bring up a lot of good points, Josh, to kind of end here about some of the societal issues and about resurgence, things like that. So let's start there next time. And then I also wanted to bring up, for those of you that might be interested, we haven't really covered so much like PPE and about like disinfectants and things like that. So we'll kind of give a little rundown of things like that about. So yeah, so we can only cover so much, right, in 30 to 45 minutes. But there's so much to talk about. All right.
Yeah, and I thought that I thought your description of some of that virology of this, I'm not seeing that other places, so I really appreciate sharing that with us.
Yeah, no, this is fun. I actually look forward to this a lot. So it's good chatting with you, Josh.
Yeah, you too.
All right. Well, take care. We'll talk to you soon.
Sounds good.